Hello again. I'm Mykola Maksymenko, and I'm Director of R&D at SoftServe. And with my practice, with my team, we actually try to help our clients and to help our internal SoftServe as a company to navigate in the future, to understand how technologies will affect their business, to understand how technologies could help them to gain competitive advantage or actually to stay alive, and sometimes to save 2 billion. So 2 billion is a lot. So try to think for a minute, what you can do with 2 million. What great things you actually, billions, what great things you can do with this huge amount of money. Because I want you to do this exercise to think what is possible and what, as a humanity, we actually can achieve when we will start to save on different aspects these huge resources that are out there. So 2 billion. Maybe someone will actually can give this later on as a question. But to give you some outline, for example, you can build a Mars rover and explore the new planet. So to understand if the new planet actually is habitable, if actually people could live there, or if we can mine some good resources there. Using 2 billion, you can, for example, build entire reproduction of a human brain, neuron by neuron, in software. So that's one of the large today's research projects. Or you can build a tower to oversee the nice Central Park in New York. So this is also a budget of around 2 billion. So maybe you have more ideas. So I would be really grateful if you could share those in the comments after the talk. But here I want to focus on a bit different aspect. So generally, this is the same sum which is needed to produce a single pill. A single pill for very basic diseases which we actually face every day. Sometimes these are some serious diseases. Sometimes they are less serious, like flu. But the research developments and all the research and developments actually take 2 billion in resources and almost 12 years or even more for R&D work. So this is a lot of inefficiency seems to be there. And what is interesting, that actually this sum only grows. So we all know about Moore's law. It's a fantastic law which gives us that each year we double our productivity in terms of computing power. So we get better and cheaper electronics, great devices. We actually get supercomputers already in our pockets. And this allows us to make our life different, to actually enhance our life. But this is completely different in pharma industry. In pharma industry, Moore's law is called Eroom's law and is inversed. So actually, each year, we spend more and more money to produce new drugs. And it takes more resources to navigate which particular molecule, which particular structure will actually become a next ibuprofen or next aspirin. So what is the complexity? Why it's so hard? And this is the picture why it's so hard. Imagine you're given the characteristic of a person in this room, for example, curly hair and some white cats and so on. So now you need to find this person in this room. Probably in this room it won't be so hard. But then you go to a city, and instead of a single person, you probably will find a group of people. And among this group of people, there will be this particular person that you look at. But in terms of a planet, this is already a challenge. So you will spend a year or two to classify this group of people, and then you spend another 3 years to actually find this particular person, to interview all of them, and to understand if that person is the one that you're looking for. So if you're talking about drug discovery, this is a very similar process. But instead of looking for a number of 6 billion people like in our planet, we actually have a number of possible molecules to search for, which is larger than the number of atoms in the whole universe. So this is an extremely large number, and this is very weird that actually we able to find any particular drug out there. So because looking for the drug there, it looks like looking for a needle in the haystack, but the haystack is now of the size of a planet. So it seems like almost impossible, and that's why it actually requires scientists to spend long time to study, first in university, then to do PhD, then to do a postdoc, and then finally they actually are able to work in the pharma company to make drug discovery, to analyze molecules while they gain this intuition, which particular structures are related to toxicity, which particular structures are related to solubility in water. And gaining on this intuition, they actually can localize the search in some area of this chemical space and spend less time. But it's still very long time. So if you look on the drug discovery process, so this pre-discovery phase where researchers actually look for the candidates, they're at the beginning. So they identify 5,000 to 10,000 different compounds and then run a lot of experiments. First, thought experiments, then numerical experiments, to actually understand which of these compounds could be successful in the real experiments on the live tissues. Then they go to and test these compounds on the mice and rabbits and dogs, cats, and so on. And only then they go into human trials. And if you go into human trials, so here we actually have 10 from few dozens to actually a few thousands of people. And imagine how hard it is to run these human trials. And imagine that actual drug that you discovered at the beginning that you thought to be successful will fail here. So you spent already 3 to 6 years for initial research. You identify, let's say, 5 candidates, and then you fail here. So you spent like 10 years on nothing, and you start from the very, very beginning. So apparently, most of the candidate molecules that pharma industry is currently working on, they actually don't make it to the final. More than 90% of the molecules in drug candidates actually fail here. And this is why we have this cycle which costs $2 billion. So that's a lot of money. And this is something that we can optimize. So currently we live in a very exciting time. So we have so many cool technologies out there. And these technologies change our life exponentially. So we see quantum computing, we see artificial intelligence, we see bioinformatics and robotics. Even extended reality allows us to gain efficiency in many aspects. So in this talk, I will show you how to use AI and extended reality to shrink this cycle of drug discovery. And in a few years, we already will see first results of these kinds of researches. And already this week, we had really cool result by one of the group in the world. And I will show it in a moment. So let's learn how AI can allow us to discover drugs fast. So as I just told you, so this week there was a really great example of that. So how fast it could be. So I just told you about 12 years, right? So this week, a group in silico medicine just showcased that a drug can go from a pre-discovery phase to actually running clinical trials in 46 days. So from 6 years, they shrink it to 46 days. So this how much efficiency we can gain with these technologies. And in a moment, I will showcase you how we can actually gain that. So AI can help us in many aspects. So we know that pattern recognition is one of the amazing features of AI. So those patterns which are usually recognized by the human now can be recognized by a computer and with a very high quality and even better than the human in many aspects. We have amazing predictive power. So now a next move in a complex tree of possibilities, for example, if we have a drug molecules, we need to understand how to synthesize it. So now we can use the same algorithm which is used for playing game of Go to actually find the retrosynthesis structure of how to synthesize particular molecule. And in this particular talk, I will focus a lot on generative adversarial networks or generative models. So you see these pictures of people. They look very realistic, right? So how many of you think that these people are real? Just raise your hands. So these people are real. And okay, how many of you think that these people are fake? Okay, actually, yeah, you have a good understanding of deep fakes images. Right, these are deep fakes. So these people actually are not real. So please volunteer out there on the R&D stand. We will use you in our experiments so you could label the data. That's very nice. So generally, in our lab, we also work a lot on deepfakes. And right now, what we do, we take facial expression of people and transfer them on a picture of different people. So in that case, we could actually produce a video of a person by just transferring a facial expression of an actor. But imagine instead of the facial expressions, we say that we want to generate a molecule, and this molecule has particular properties. So instead of these nice-looking pictures, we will generate nice-looking molecular graphs. And as I just said, that we have many applications of machine learning, and here is a map that we created at the beginning of this project, so where we can actually apply our knowledge and our skills to enhance drug discovery. And generally, there are many of really cool examples of those, but most of them are not as revolutionary, as exponential. So we really wanted to focus on this generative abilities of neural networks, because if we could, at the very beginning of our drug discovery cycle, discover the right molecules with the right properties, we could actually allow experimentalists to go the way to the clinical trials much faster. And that's what the group in silico just did as well. So what does generative network allow us to do? So imagine we had this large space of possible molecules. This is gigantic. It's impossible for a human to actually even label all these molecules or even try half of them. But what we can do with these networks, we actually can segment this space into parts, different parts. And different parts, like here, the colors, will actually represent different physical properties. And in that case, we would then say, okay, I need the molecule with this amount of solubility. So then I will look in this part of the space, take some point in this space, and generate a molecule from that point. So now, instead of looking for a needle in a haystack, which is a gigantic haystack, so it's impossible to find anything there, so we actually generate these needles and generate as many as we wish. So what will be needed from us further on is just like quickly check if some of them fit our requirements for a particular drug. So to do that, we actually employ a generative model. And here is one example. I will not show you all the zoo of those generative models. There are many of them. And people just compete with each other whose one is better. But this is the most simple one. And we actually showcased in our study that you don't need anything much more complex. So actually, your model could be simple enough for production needed. So what it does, it actually has a database of drug-like molecules. Let's say we have 6 million of molecules out there already publicly available. And what we want to do, we want these neural networks, which are in our model, to first shrink the initial representation of this molecule into low-dimensional space, and then actually decode from this space back into original molecule. So when we train this model, we actually want to get something as input and reproduce this input. So in this way, actually, the model learns to build this map, as I told you. So instead of having this very complex graph representation, we will have some kind of few-dimensional map. And in this map, each point, it now will be continuous instead of discrete. So this one is discrete. Here we have continuous representation. And now we can continuously just change go around this map. And each point in this map will represent a new molecule, which was not there in the training data set. And important fact is how to represent the molecules. And there are a lot of different cases. So one case is just take this original graph as image. Another case is take the graph representation as I just showed on the previous slide. But we used one interesting point that in chemistry, people actually use a lot of simplified molecular input, line entry specification. This is specifying the specification of a molecule as a string. So what it gives us, it actually allows us to treat this string as a language. And I will tell you a really great example how we use it. So at the end, actually, our generative pipeline looks the next. So we have this SMILES database of molecules. This is a large database. And it passes through this encoding and decoding pipeline, so our generative pipeline. And at the end, we want to decode it back. And by the way, here we have also another neural network which predicts the properties. And we have another really cool thing here, which is so-called spell checker. So it's the same thing that you use every day when you write email to someone, we use for the molecule as for the molecular language. So at the end, what we do, we actually generate then these points in that representation and pass them back via decoder to decode the molecule and predict the properties immediately. And then by looking at these properties, we could filter out unnecessary stuff, and this is done in just like a fraction of a second. And see if the graph, that resulting graph that we got actually passes some filter here. But it's also important to say that when we just randomly take some of these points in this map, so it does not guarantee that this molecule will actually be physical, if this graph actually will make any sense, because this is not something present in the database. So it was very important to create a check if this line representation actually makes sense. And I also wanted to say that one particular mistake in this line could actually be a small mistake for the neural network, but it could lead to completely wrong graph, wrong molecule at the end. So what we created a spell checking model, which is based on the same model that we use, for example, in Facebook or in Grammarly or wherever. So it's so-called attention-based sequence-to-sequence models, which actually takes these strings as input and try to correct missing parts of it. So for example, here we did not have an open bracket. So after passing this model, it actually put here the open bracket. So that's the idea. And what we've seen that almost 67% of the unique molecules that we created actually were corrected by this system. So this is very important in our pipeline. And by the end, we also looked if these actually new molecules that we created makes any sense from the physical representation. So we checked actually their properties. So we took some of the parts of these molecules and checked if the distribution of these functional groups, these structural elements, is similar in the resulting database or it's very much different, because we want to keep the distribution more or less realistic. And one other experiment that we did that no one actually tried so far is instead of running immediately from this generative model to the laboratory and try it in experiments, we actually created another check, which, for example, checks the property of solubility and runs a numerical experiment on these molecules. So we created 1,000 of these molecules in some finite space, virtual space, and checked the properties like solubility or some other properties that we can actually focus on. So just to give you an outline, I did not dive too much into details here, but we could use AI to speed up drug discovery a lot. And we already see first results in this space. So what we did in our research, we actually checked if we can use small databases instead of large databases. Because in real life, actually, these pharma industry laboratories, they actually are very much separated. And each lab has not so many examples of right molecules with the right property set. We also built this really cool example of structure spell checker. And at the end, we also used molecular dynamics as experiment. So now it's much more probable that when we go to the real life in vitro experiments, that these molecules actually will make sense. So we more or less covered this 3 to 6 years with an algorithm which actually works within a few seconds. But we did not stop here. So actually, as I just noticed, this example was that instead of 3 to 6 years, we still have 46 days. So there's still a lot. And this is because different groups on these different structures here, for example, in drug discovery, preclinical, they actually talk different language. So once scientists come up with a molecule, they go to another group. This group runs experiments. Another group actually runs numerical experiments. Some other scientists come in and say, oh, look, this structure does not make sense. So there is actually a lot of collaboration between scientists. And this collaboration in the past was done on this kind of models. So people actually build these models, analyze their structure, because it's very important to know what is the spatial structure of the molecule, such that it fits particular binding site of a protein that it should block. Then actually people moved to a 3-dimensional software where you can see these models actually on the screen. It's nice, it's much more efficient, it's much more faster, but now actually the spatial component is lost. And also collaborative component is lost. So people don't collaborate that much now. So we created a tool which actually allows to visualize these molecules in AR, augmented reality. And this is the first AR tool in the world which actually operates with molecules. And what it does help us to do. So first we take our molecules from neural network, from AI, and create actually a variety of them in your space. So you can go around, see these molecules, and take the one that you like the most. Then you can actually analyze the structural properties of it. See what is the distance between atoms, what is the angle between planes, what is the angle between different 3 atoms. And this, moreover, this can be seen by different scientists in different glasses at the same time. So the group of scientists can actually collaborate and discuss particular structure. So there is much to do more, of course, here, but this is a really first example of AR. So I'm very proud of our ideation session that we actually came up with this solution. And let me actually showcase you a really cool concept video that we created after we ideated this.
Start to finish, a new drug can take 10 to 15 years to reach the market at a cost of over $1 billion. Screening of drug candidates is a long and complex process. Artificial intelligence allows to directly generate new molecules with desired property without a necessity for long and costly search. Extended reality allows to access structural property of the new structures and understand interactions between the molecule and a target. This solution allows teams from different departments to work more effectively together. Are you ready to visualize improved drug development?
Okay, did you like it? I really want you to cheer this fantastic group of people who actually help us work on this project. So this is a life science research team, Max, Dvinka, and Alex. Alex is actually somewhere here. So you can really talk to him a lot. So he's a biochemist. I think he is there. Yeah. And we also have Zoriana and Pavlo here. Vitalik and Nick, they work on this XR application. We also have marketing team who helped us a lot with making this nice looking video. So please cheer these people. And what's next? So this was our first step in this dimension. So it's still a lot to do. So we have first example of AI helping drug discovery. But there are a lot of new things to do. So one thing is that we work with small molecules so far. It's still a challenge to model large molecule, and particularly for peptide molecules. So in our lab, we currently focus on how to approach this problem. Another problem is that even small molecule is much more efficient to run immediately on the quantum computer instead of running this large-scale molecular dynamic simulation. So our focus is actually on developing expertise in building quantum computing simulation of small molecules. The future is in robotics for automated drug discovery. So instead of scientists working in the lab and ideating on the molecules, we'll have generative models ideating, then we'll have a reinforcement loop. You learned about reinforcement learning just a few talks back. We'll have a reinforcement loop which actually will learn to correct these molecules, and then we will have a robotics lab which could immediately synthesize these molecules and check the properties. So this will be really a huge advantage, not only in drug discovery, but also in general material science. So we could create new materials for solar energy, for new sensors, and generally for new type of objects that could, or coatings that we need if you, for example, want to go to space. So sensing and interfaces, this is something that will be changed very, very soon. So mind the future. Thank you.
Thank you, Mykola. Thank you. So now it's time for the questions. Let's pull up our Telegram channel. Let's pick up the first one. When did that project started? Which one?
Okay, so generally we started to look into the life science at the end of the last year, around December. So we got the first results already in January, and more or less the first concept of the paper by March. So the paper is still refined because it's like 14 pages, it's a long process, but yeah, so that was the main research. And then we actually ran another part which is AR and augmented reality stuff. So that took another few months, but it was rather quick. So this is on the early stages of development. So not so many people actually work on that, as you've seen.
All right, so the next question. What do you think? Is it possible that commercial pharma companies merge their forces and collaborate to develop important life-saving drugs?
So generally there is this collaborative effort, but it's very hard because the IP, intellectual property, is something that these companies actually worry about a lot. So they don't share data that easily. And there are a lot of conversation of how to build a platform for sharing data, for sharing this experimental data on what the property of particular molecular graph is. So it's quite hard. So there are a lot of bureaucratic issues, but there is a lot of effort as well. And what is happening right now, that there are a lot of these smaller groups emerge of scientists who try to use advanced technologies to speed up drug discovery. And that's where actually a lot of collaboration between these small companies and pharma is actually happening.
All right, so the next question. Is it possible to use the same approach to generate new materials such as food, etc.? What difficulties can prevent from that?
What difficulties? Okay, so as I just said, that material design is similar, it's just identical. So our paper is actually called Using AI for Drug and Material Design. So it's the same procedure, it just like drug discovery sounds a bit more sexy, but the procedure is the same. And we could really discover new materials, for example, for solar energy, which is really important of how to build new kind of batteries to accumulate solar energy, or new kind of elements to actually harvest this energy. So this is what actually another line of research is focused on. And what difficulties it can prevent from that? I don't really understand this question.
All right, let's pick up the next question. Can you say a few words about IBM Watson system and its capabilities in this area? I think life sciences in general.
Let me not comment on IBM Watson. So it's actually not a competitor here at all. So it's more a generic purpose system with very like shallow algorithms without no very deep expertise in that. So if you want to use IBM Watson in this area, it's no way.
All right. So I think I can comment myself on the next one. So why is the ML called fusing here? So it's basically not a fusing machine learning. It's rather how we fuse machine learning and virtual reality. So that's the right way to think about that. So the next question, do you have any competitors in this field?
Yeah, so there are actually a lot of labs, as I say. So there are competitors and collaborators, of course. So currently we talk to different groups in this field, and we establish this trust to build new next researches in collaboration with them. And these are groups, for example, in Cambridge, in ETH Zurich, various quantum computing companies as well. So we try to be very open. So we are not a black box in R&D. We actually try to talk to other people and do this in format of open research. And what else was there? Drugs from cancer.
Right.
So can we discover drugs from cancer? Yes, we can. So this is one of the aspects, and probably a very basic example of how can that be done. For example, you have a protein, a gene, which is responsible for a cell to actually divide, right? So if there is this defect gene, so there will be some cell which will be spontaneously dividing, and actually there will be a cancer part in your body. So the important fact here is that you need actually to find the right molecule, the right drug, which will block this gene. And this is exactly how you can use the same approach.
All right. So how long does it take to create a 3D model for a device? Wouldn't it be too expensive?
The 3D model actually is generated on the fly. So the output which we get from neural network is a graph structure, and then we have a script which actually creates the 3D model. It's literally a fraction of a second, so it's very fast.
Cool. What did inspire you to work on that project?
What?
What did inspire you?
What did inspire me? Well, this is a cool project. We love healthcare. We do a lot of healthcare at SoftServe, and you've seen that really cool stuff which we did on biosensors. And because I'm myself material scientist, that's how the innovation actually comes from.
All right, so the next question is, the same approach can be used for human evolution in the future?
For human evolution, that's a very hard question. Well, I think yes. Again, targeting specific genes, you can do a lot of cool stuff with your body.
Okay. So what programming tools and languages did you use?
If you talk about AI pipelines, here the pipeline is built in PyTorch, which is a very general and but at the same time specific framework for building machine learning models. So you can do a lot of things. It's very flexible. And for this backend, you don't need anything else actually. So you can run it in TensorFlow as well. Then for the front end, the guys use Unity for these nice looking 3D models. And that's also a general framework for building applications for AR and VR.
All right, so I'm going to skip the next question because narcotic drugs are illegal. That one, other drugs which were developed by your app and which are already used in nowadays medicine?
No, not now. So actually that's one of the aspects that we currently elaborating and searching for collaborations in the laboratories in universities, such that, for example, we create a bunch of molecules. Can these molecules be tested in the real wet lab experiments? So from the medical point of view, this can be done. Then also it's interesting from the material science point of view. So we also talk to some laboratories from the material science perspective. So yes, we work in this direction, but we don't have anything done yet.
So we still have some time to take a few more questions. So how do you see this project in the future? What are your plans?
Well, that's what I just said. So there is a lot of room to explore. Explore material science, explore different properties of materials, explore better models, which could actually generate these molecules without much errors. Explore large molecules. So if it here use generative models for generating small molecules, for large molecules it doesn't work. So we currently explore, for example, reinforcement learning to find the right conformation of the large peptide. Then again, I said the quantum computing, that's a very important field to navigate in, to understand how it works. There are a lot of customers in the world who are already working on material science with a focus on running experiments on the quantum computer. So this is something that we also try to lift our expertise at SoftServe in.
All right, so maybe the last question, just to give some takeaways to the audience. So can you recommend any resources or books or materials to read or watch about what we've just discussed?
Any resources? So, well, there is quite plenty of lectures. And these lectures are both on academic side and in like public blog posts. So some of these databases are open. So you can really go in, try your own model, which will generate molecules with particular properties. But if you want to go into deeper research, of course, you need to build these trusted collaborations with some universities, some research groups, because then you can do much more. Not something beyond what is available in the open discussion on the forums.
All right, that was the last question. So thank you, Mykola.
Thank you.